NAEC San Diego, speaking about Ostreid herpesvirus 1, I guess there's more than one, infections of the Pacific oyster and Tomales Bay. Is that right?
Yes.
Very good. Pointer. You see it? It's gone.
What do you need?
I was looking for the pointer for you, but I can't find it. You don't need?
No.
Good. So you have on it.
You want to pop this open for me?
I just... That good? Or you want to make it bigger?
Well, if you hit the, blow the whole screen up and get rid of all this stuff.
We can do that right off. Well, let's just see if that works. I don't know. It's got a flat tire.
Okay.
Sorry.
Can you hit the little thing there?
Do you just want it bigger?
No, just hit the display. Yeah.
Okay, so if you look over there,
Okay, gotcha.
what you're seeing
Okay,
is the-
perfect. Okay, thanks.
Yeah, you're good.
Okay, quick question for you. How many people in this room have heard of an Ostreid herpesvirus? Well, that's very good. I'm pleased. So what I'd like to talk about now is the original Ostreid herpes virus that showed up in Tomales Bay around 1993, and then a new variant that's particularly concerning because the original one killed oyster seed, and other than purchase, growers don't have much invested in oyster seed. The new microvariant kills larvae, seed, juveniles, and adults, so it's particularly concerning to the global oyster farming industry. I'd like to mention commercial partners in this, Hog Island Oyster Company, Baywater, and Taylor Shellfish Farms. On the left-hand side, you can see Pacific oysters, and that's our primary goal. Tomales Bay is right in the center as the backdrop there. It's a relatively small bay, about 40 miles north of San Francisco, about 10 miles long, and averages about a mile wide. On the right there, you see an electron micrograph of some oyster tissue. The circular image on the left-hand side of that is the nucleus. In the lower right-hand side, the speckled is a membrane, and what looks almost like a black pearl necklace strung between those two are viral particles. I'd like to also acknowledge Carolyn Friedman and Colleen Burge, and this is really their presentation with results from a Sea Grant funded project that they had. I had a fairly small role in the original project, and I'll talk about my larger role in another project that was just funded. If you get a little confused because I'm talking about things that have been done and things that are proposed, that's why. This is kind of a hybrid presentation on two research projects. Carolyn's at the University of Washington. Colleen Burge was her graduate student who started in 2002, has since gone on to be an assistant professor at the Institute for Marine and Environmental Technology. We've got some collaborators at UC Santa Barbara, and I'm with California Sea Grant. So the main aim of the proposal is to give the shellfish industry options and tools such that if this new microvariant herpes virus shows up, they have some options with how to deal with it. My primary role in the new proposal will be to provide educational materials and training to help prevent spread of this new microvariant. And the audience for that is really large. It's shellfish growers throughout the country, it's resource managers throughout the country, it's extension agents throughout the country. And so that will be something that will involve a whole lot of people. In addition, we need diagnostic tools. We need rapid assays for this. Very, very important is to identify resistant oyster lines and perhaps even other species and determine the relative risk of transmission among these different life stages. Real quick, importance of oyster culture. On the left-hand side of this is the United States production. The U.S. West Coast leads with about 100 million in production, followed by the East Coast with around 50 million and the Gulf Coast with around 24 million. The Pacific oyster is the primary one, and the goal of this project is to address some of these emerging diseases. The graph that you see on the right-hand side, you can barely see the production levels for the European oyster or the Olympia oyster. The total production is the red dashed line, and the Pacific oyster is the green line. You can see how closely they track, and that's just further evidence that Pacific oyster is really the primary mainstay of the industry. So short pulses of mortality of Pacific oyster seed started in Tomales Bay in about 1993. Up to 95% of seed died. Over the next 10 years, we looked at every possible environmental variable that you could to try to determine what was causing those mortalities. We were not able to do that. We looked at environmental toxins, we looked at algal toxins, we looked at different physical variables, we looked at pesticides and herbicides that might be coming into the bay from the surrounding area, but it's mostly an agricultural dairy area, and as I said, we were not able to find out what might have caused that. The oyster herpes virus is something Carolyn continued working on, and she first detected it in 2002, and then looking at archive samples, traced it back to as early as 1995 in Tomales Bay. I suspected it was there in 93 just based on the mortalities, but there weren't samples to confirm that. It's not culturable. It's a really difficult virus to work for in that regard. The large mortalities of seed that the oyster growers saw caused some to go out of business, some to relocate. But fortunately, we were able to work around the problem, having identified a season where you could plant seed and you wouldn't suffer those mortalities. And that was basically, you don't plant seed after April and you don't plant seed before October. And if you do that, then mortalities, instead of being 60 to 90%, are down to 20 to 30%, which is pretty standard for the industry. It's not going. Oh, there we go. So let me talk about a little bit about international distribution of the oyster herpes virus. In the early 1990s, it was in France and New Zealand. 93, it was found in Tomales Bay, California, on the west coast of the United States. Around that same time, it was found in Japan, Korea, and China. Moving forward to 2008, 2016, you can see the development of this new microvariant indicated by the blue stars. It too was found then in Japan and in China. It showed up in Australia. It then moved down to Tasmania. It showed up in New Zealand. It showed up in Europe and spread pretty much throughout Europe. There's a few countries where it wasn't, but for the most part it spread throughout Europe up to the Netherlands, Sweden, and Norway. In Ireland, in 2008, it was in 3 of 30 of the oyster culture bays that they have. By 2012, it was in all 30 oyster culture bays in Ireland. Quick project background. As I said earlier, the ability to kill seed and adults really heightens concern about this. Fortunately, some resistance has been shown in oysters that have this virus in France. And hopefully these resistant oysters are able to both limit their infections but also to expel the virus from their tissues. In New Zealand, oyster susceptibility to the U-bear decreased over time, which is another good signal that there might be potential for providing disease-resistant stocks. So it looks like that's a promising avenue. In 2016, at the PCSGA Shellfish Health and Disease Panel, this new UVAIR was the major focus of interest and of questions. So Carolyn and Colleen put together a proposal that was funded by the National Sea Grant Office to do some additional work. Real quickly, some new data that came from the first Sea Grant funded proposal looked at the body burden of OHV-1 per oyster. And on the right-hand side, you can see evidence from 2 different selected families that were compared, 32 by 52 and S58 by 19. 58 by 19 is the green. The viral load on the left-hand side, you can see, is different between those two, once again giving encouragement to the idea you might be able to do some selective breeding for resistance. In oyster survival on the right-hand side, you can also see the 58 by 19 had better survival than the other family. So this is all encouraging. So to look at a little model in Tomales Bay, you've got a temperature effect. It's another thing we discovered. It appears that this virus is not virulent at temperatures below 24. I'm sorry, yeah, below 24. Above 24, it becomes virulent. You then have viral mortality that's induced. Some things that we don't know, though, are what are the role of alternative hosts? Are there other hosts that could be providing a reservoir in the bay that would result in recurring infections? We don't know a lot about disease resistance. We know there appears to be some, but that's something we need more information about. And we don't know really anything about host physiology and susceptibility and how that plays into disease resistance. These are some results of a study that was done by a group of researchers in Australia. And the only thing that I want to point out here is that on the left at the top, you can see low concentrations of viral particles. The concentration of viral particles increases as you go down that left axis. But I want to point out the timeline at the bottom. So from 0 to 48 hours, you've got extremely high viral loads in these animals because it's able to replicate so quickly. And these would likely be mortality unless you had oyster stocks that had considerable resistance. So with the new proposal, we've got three major hypotheses. One of which is that species and family lines will demonstrate differential survival and allow us to selectively breed resistant stocks. That the families resistant to herpesvirus 1 are also resistant to herpesvirus 1 microvar, and there is some preliminary evidence out of France that that's the case. And this is really promising news because there's been work to develop lines resistant to HV1 for many, many years. The other thing that we want to look at is genomic variation of viral strains to see if that variation genetically can explain difference in virulence. The objectives, one of the primary ones, develop a multiplex real-time qCPR so that when you suspect it may be present, you've got a really quick, objective way to find out whether that's the case or not. Conduct field trials to oyster families and species with differential susceptibility to document that. Laboratory trials to examine differential seed survival to the HV1 microvariant. And to sequel the viral genomes used in infection trials with the California strains. So conclusions that we've got are, yes indeed, HV1 and its microvariants are serious pathogens of oysters. Our goal is to limit spread by education and outreach. This is really a goal and an objective, and that's the primary role that I'll play in this project. And then Carolyn will work to aim to develop these molecular assays in oyster lines to better enable management of the disease, early detection, and adaptive management of the oyster stocks should the disease show up in North America. Thank you.
Questions for Paul?
Is there anything, because of the temperature, 24 degrees, is there anything an oyster grower could do on a practical basis during the summer months, like with a slow rotating paddle wheel, or to circulate water, to bring up cool water from somewhere to? Probably not.
I can't imagine. Most of the oysters grown, at least in California, and I think all along the eastern seaboard and in the Gulf, it's really shallow water. There's not deep cold water nearby, and I don't know that would be viable anyway because it's spread over such a vast area. What you can do is exactly what they ended up doing is don't plant after April and just kind of adaptive management like that.
Are there any regulations preventing it, or do they check?